Okay, so that's the simple cell. Anybody think this is the cell simple now? No, not hardly. All right, so we're getting back into where we left off. Sorry for not being here next week. I would have much rather been here than where I was at when I was doing last Sunday. So I'm glad that I'm back here this Sunday to continue on with our lesson. All right, so why do people challenge Christianity so much more than all the others? Anybody know? Huh?
Because it's true.
Because it's true. That's a good answer. Absolutely. Any other reasons? Why is Christianity challenged? I mean, you don't really see Islam in debates on TV and stuff, and Hinduism in debates and things like that. Why is Christianity challenged so much? Why is it the one that really seems to be challenged? Why is that?
It's the dominant religion here.
Here, maybe in the West, but it's certainly not the dominant religion across the world. But here. Any other answers? Okay, think of it like this. It's really the only worldview that's open to this type of assault from other people. Okay? It can take on these tough questions and other things, that other worldviews that people put up against it, and has answers for it. Okay? It's pretty important to know. When you think about it, that ought to tell you it's true. Okay? That ought to help you with that. Okay? This past week I came across a news article. I was going to show you the news article, but I'm having a hard time getting the internet feed here and everything. But it was from Fox. And remember a couple weeks ago we talked about ice core drilling. Well, this particular, I mean, this is current. This was in the news just this past week. And it talked about they were doing this ice core drilling and went down like a mile, 2 miles, something like that. And guess how long ago they said they found a sample of a microbial. Guess how long ago it was from? Millions of years. Okay, just like we talked about a couple of weeks ago, how they'll say that it's millions of years. And they suggested that. And then they also found this microbial, and they suggested that it was the secret to life. Well, we know the flaws for different dating techniques. We've talked about it, man, ice core drilling. But here's a thought for you. If they can't figure out the secret to life with fully formed and functioning human beings, what makes them think they're going to find the secret to life from a little microbial they find underneath all that ice? You ever thought about that when you hear these things on TV? Think about that the next time, okay? The secret to life, we can find it. It's on Mars. We found this little organism. Really? We have all these humans walking around and we can't figure out the secret to life. How are you going to find it from an organism from Mars? Anyways, a review. I just thought that was an interesting news article. These things trigger my brain sometimes when I read these things. Hopefully they do yours too. If not, think about it from an apologetic standpoint. Review, a couple weeks ago. We looked at what reliable evidence shows. Keyword reliable, okay? Keyword. Not assumptions, not deception like in this news article. And we looked at 4 dating techniques, 4 dating methods. What were they? Anybody remember?
Geological strata.
Geological strata dating. Carbon dating. Who said it over there? What else? Radiocarbon dating. What else? What else? That's 2 of them. We talked about 4 primary ones.
Radiometric dating.
Huh?
Radiometric dating.
And what else? Now, what was the 4th one? I just talked about it.
Ice core.
Oh, check that out. Ice core drilling. Very good. Good catch, Lee. Okay, so we talked about those 4. Geological strata dating. There was a fallacy with that. What was the fallacy with the geologic strata dating? What was the fallacy?
They use the rocks to date
Say it again.
the rocks.
Say it louder.
They use the fossils to date the rocks, and the rocks to date the fossils.
So that's called what?
Circular research.
Circular reasoning. Very good. Excellent. Circular reasoning. So if the ages of the fossils are used to date the strata, then the strata can't reliably be used to date the fossils, and it's circular reasoning, and those are unreliable. If you hear somebody using circular reasoning, it is unreliable. Carbon dating. Carbon dating. Actually, I just gave you the answer. What was the other? How can you do it in the reverse? What was the real name for carbon dating?
Radiocarbon.
Radiocarbon dating. Who said that over here?
I did.
Okay, get up and I'll see what it is. Okay, so in short, they're assuming how much carbon is there, and then when something has died, and then they compare how much has decayed, okay? They make a comparison between the 2, okay? Then radioactive carbon has an expected life of less than 62,000 years. So using this method to arrive at millions of years, it's not even scientifically possible. Not even possible, because the life of it is only 62,000 years. In other words, they are assuming, making blind assumptions. Okay, so watch out for those. Also, the older the sample, the more unreliable it is. probably that it's going to have higher contamination. Many examples that I gave you of some of these flawed techniques. Does anybody remember any of the examples? Any?
Mollusks, yeah.
Huh?
Plane that went down and they dated with the ice core.
Yeah, it's called, what was that called?
Some plane.
That was called the Lost Squadron.
Yes.
Okay, during World War II, it ran out of gas, landed on Greenland. They went up back and excavated it 48 years later. It sunk down a certain point, and they said that it was supposed to be, let me find my note here, 1,824 years old. But they just excavated it 48 years later. So we know ice core drilling techniques are very, very flawed. Any others from another?
Living mollusks?
Huh?
The living mollusks?
Yeah, how long ago was it? How old was it?
They dated as 2,300.
2,300 years old, and it was living. That's a pretty old mollusk, I would say. Okay. Another example, 1971, a freshly killed seal. Freshly killed. It had just been killed. Was carbon dated as dying 1,300 years ago. I'd say there was a problem with that. Okay. Another one that I gave you using the isotope potassium argon. Okay. Mount St. Helens eruption. They said that it was dated at 300,000 years old. I was alive when that happened. I don't think I'm 300,000 years old, okay? Just so you know. And then, of course, the ice core drilling techniques. We also looked at the Grand Canyon. What was the issue with the Grand Canyon that we said there's no way it could be millions of years and it was probably from the flood? Huh? That's right, but what was it called? Remember? How do you say it? Called the Khabib of
Khabib,
yeah. Very good, excellent. So it shows where the slopes to the top of the Grand Canyon near the lower end of the river. It slopes up 69 to 8,500 feet in elevation. Okay, very good on that one. We looked at some young Earth evidences. Anybody know what some were?
Galaxies line themselves up too fast.
That is a universe explanation. Very good. So, okay, I put that in there just as review from last
Not
time.
enough mud on the seafloor.
Not enough mud on the seafloor. That's a good one. Very good.
Not
Any
enough
others?
sodium in the sea?
Sodium in the sea. Very good. So realize with the sodium, there's many other elements in the sea that calculations give much, much, much younger Earth ages. And the flood answers it perfectly. It was perfectly the flood. What's some others? Anybody remember any others?
Coriolis effect?
The Coriolis effect. What was that?
It's slowing of the Earth's rotation.
Say it again.
Slowing of the Earth's rotation.
But what is it? What does that cause? Here, I'll tell you what it was. It causes desertification. Anybody know what that is? Now that I've told you. It's where the deserts are expanding. Okay, the deserts are expanding. So the Sahara shows that at one point there was no Sahara, and it's expanding a little bit each year. When the dating of the Sahara goes back to 4,000 years. What happened roughly 4,000 years ago? Huh?
Flood.
A flood. Very good. Then we talked about what came first, male or female. So evolutionary theory predicts nature would be asexual rather than sexual reproduction. So how could complex sexual diversity arise or even survive at all? We looked at that a little bit. So this week, we're going to go into, we're going to use a couple of videos because, again, I want you to hear from experts themselves and what they have to say. They're not long, 8 minutes or so, each one of them. But pay attention because this is part of the lesson, okay? Besides that, you'll get a break from me talking all the time anyway. All right, so the first one.
Natural selection acts
You hear it?
only by taking advantage
Everybody
of
hear it?
successive variations. She can never take a great and sudden leap, but must advance by short and sure, though slow steps.
I'm a scientist, a biochemist, and biochemistry studies how life works at the level of molecules and also how life came to be the way it is. The standard answer for years has been that, well, we sort of evolved gradually by natural selection, but there are large problems with that.
Since 1988, Dr. Michael B. has investigated complex biological systems that seem to defy explanation by natural selection.
For the longest time, I believed that Darwinian evolution explains what we saw in biology. Not because I saw how it could actually explain it, but because I was told that it did explain it. In schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution, a Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before. And the arguments seemed pretty convincing. And at that point, I started to get a bit angry because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and since have decided that Darwinian processes are not the whole explanation for life.
Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
There are as many molecular machines in the human body as there are functions that the body has to do. So if you think about hearing, seeing, smelling, tasting, feeling, blood clotting, respiratory action, the immune response, all of those require a host of machines.
When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
Many of Behe's doubts were tied to a remarkable biological motor.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts and all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed. That's no chance assemblage of parts.
Behe's reaction was not surprising. For the molecular motors that drive bacteria through liquid, each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force, it has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's, it's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation.
Darwinism was a lot more plausible when we were thinking about globs of protoplasm than it is when we're thinking about molecular machines. Scientists in Darwin's day were hopeful that the complexity of biology would resolve to simplicity once they found the basis of life. But we found out the exact opposite. And in the 20th century, we found out that the lower you go from whole animal to organ to tissues and cells, the more complex life becomes. And at the very basis of life, the cell, is just outrageously complex. And so the 19th century hope that life at the bottom would be a simple phenomenon is spectacularly wrong.
All right, Darwin thought the cell was simple. Is that simple? No, far from it. Thank you, Jake. Far from it. Okay, so we saw a video like this a couple weeks ago, but not near as in-depth as this one. Darwin's microscope only magnified about 200 times. Today's microscopes can magnify like 50,000 plus times. So a huge difference. So the more science advances, the more it shows that the Bible is right. Realize that. Some people say, well, we don't want science to advance. What might it show? We might find aliens out there or something. No. The more science advances, the more it shows, hey, the Bible had it right all along. Keep that in mind. One cell is incredibly complex. Even today, computers cannot fully comprehend just the cell, even with the computers that we have today. In the DNA alone, there are 3.1 billion letters of DNA in one cell. That's a lot. Let me read you 2 verses. Okay. Isaiah 44, 24. Thus saith the Lord, thy Redeemer, and he that formed thee from the womb. I am the Lord that maketh all things. Then Isaiah 49:5. And now saith the Lord that formed me from the womb. So next time you read these verses and verses like this, think about some of these things we're talking about, okay? It's not Darwinian evolution that caused it. Single cells are high-tech factories, complete with artificial languages, decoding systems that have central memory banks that store and retrieve huge amounts of information. They're very, very complex. They have precise control systems that regulate the automatic assemblage of parts and components. They have proofreading, quality control mechanisms that guard against errors. We're talking about one cell here, is what we're talking. They have assembly systems that consist of prefabrication and modular construction, complete replication that includes every living cell, every living organism, and it duplicates itself with bewildering speed. We cannot, mankind cannot duplicate what the cell can do. Even with all the technology we have, we're not even close. Okay? And all that is the size smaller than the head of a pin. So we're not even close what the cell does. Okay? So Darwin was completely wrong. It's one of the most complicated things on earth, the cell. Scientific Journal, now this is from a science journal itself, said for one life to be viable within one cell, one simple cell, it would need to have 239 protein molecules, each containing 445 amino acids, which are made up of 10 to 20 atoms. And out of the hundreds of amino acids, only 20 are used in the protein, and they can't simply float around randomly. All of the aminos have to be lined up in a single line in perfect sequential order to have a protein to function. That's not the cell, that's just for a protein, okay? In other words, in order for life to happen, these aminos would have to line up perfectly, not once, but they have to line up perfectly 239 times to form 29 proteins, which would make a living cell. Okay? We're talking out of hundreds. They have to have the right ones, and it has to happen 239 times perfectly in perfect sequential order. Okay, we'll be seeing a little bit more of that in the next video that we're going to show shortly. Okay, this is why even some non-Christian scientists are abandoning Darwinian evolution. They're saying that this simply can't happen by chance. That's kind of what Michael Behe is saying. That's who you heard from just then. He said this can't happen by chance. The probabilities we talked about are huge. So even if we had billions and trillions of years, it just simply could not happen. And that's just one cell, let alone trillions of cells in the body. Not organs, not a person fully formed and functioning like we are, let alone all the species out there today. That's just one cell. Charles Darwin, in his book, The Origin of Species, his famous book, he said, by his own admission, quote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modification, my theory would absolutely break down." He even admitted that. Well, guess what? It's way past broken down. Way past it. This is why I gave the statistic in week 1, this one right here. For one functioning protein, Doug Axe said the correct sequence of aminos to get one functioning protein, the odds that mutations can cause a functional combination, hopefully you remember that. I haven't erased it. I haven't erased any of these from week 1. That's the reason why. The odds of it happening is 1 in 10 to the 74th power, or 1 chance in 100 trillion, 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 trillion. And it would be like the example of blindfolding a man and having him search the entire galaxy for an atom. That's the equivalent of that number right there. That's the equivalent of having a protein come into existence on its own by random chance, okay? So a couple of weeks ago we talked about mutations. Does anybody remember that? Mutations talk, like week 2, I think, maybe? We're going to talk about it a little bit more in depth because there's a whole lot more to it. We just touched on the surface. Mutations are accidental changes in the genomic sequence of DNA, and that's not good. That's not a good thing. Because human mutations generally result in relative weakness or even premature death. But in spite of the flaw, this is critical, absolutely critical to modern macroevolutionary theory. Macroevolution. Remember what we talked about in week 1, okay? So science also will look at examples of adaptation within species. Remember how I said you will see examples of microevolution and then they'll roll in macroevolution, okay? Are species and like kinds the same thing? We talked about this already. Are they the same thing? No, not at all. Okay? Keep in mind, like kinds are not necessarily species, and a like kind could be several species. Okay? Remember the example of the 3 things? I said which one does not belong, and y'all said the banana didn't belong. Remember that? And the other 3 were a wolf and a dog and something else. Well, those could be different species, but they are of like kind. Okay? So don't get tripped up by this, because they'll try to get you on that one. Do you believe in evolution? That whole question. So an example. There's a certain strain of bacteria known as, and we'll just keep it short and simple, MRSA. In your notes it has the whole word, I think. MRSA, for example. And it might be able to adapt to resist antibacterial medication. And by the way, you'll see this more and more. It's becoming a problem for future sicknesses, and that is bacteria becoming resistant to antibacterial medication. Okay, you see it in the news sometimes. That's why we get flu shots every year and say, well, this new strain of flu has come out, and we may not be able to resist against it, even though we've had our shot and all that kind of stuff, because it's adapting. Okay, but adaptation is not mutation. Anna ran into this with a friend in a conversation some time back. I don't know if you remember, but I remember your conversation with somebody about adaptation versus mutation. Okay? Adaptation does not accidentally alter the bacteria's DNA. The adapted MRSA strand that we're talking about in this example is still bacteria. It doesn't turn into a monkey or something. Okay? So it doesn't. It just adapts. It doesn't mutate. It's very important to not get stuck into the question: do you believe in evolution? As we said in week 1, they'll use microevolution as an example, kind of like what we're talking about, and then roll in macro and say, see, evolution has to be possible. It is not. In fact, in humans, beneficial mutation is an oxymoron. Adaptions can be beneficial, but human mutations are not. Do mutations happen?
Yes.
Yes, mutations do happen. There are several thousand known mutations of human DNA, but not one is truly beneficial, okay? Every known human mutation is ultimately weakening or even deadly to man. It doesn't advance it, as Darwinian evolution would suggest. Stephen Jay Gould, who's a pretty outspoken, famous kind of evolutionist, he referred to this as the academic equivalent of murder. Okay, that's an evolutionist talking, not a Christian person. All right, so now one more video. It's about the same in length, but it's going to go into a little bit more of what they're talking about.
In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G, for adenine, cytosine, thymine, and guanine. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Like other scientists working on the origin of life, Kenyon realized he had two choices. Either he had to explain where these genetic assembly instructions came from, or he had to explain how proteins could have arisen directly from amino acids without DNA in the primordial oceans. And in the end, he realized he could do neither.
It's an enormous problem how you could get together in one tiny submicroscopic volume of the primitive ocean all of the hundreds of different molecular components you would need in order for a self-replicating cycle to be established. And so my doubts about whether amino acids could order themselves into meaningful biological sequences on their own without pre-existing genetic material being present just reached, I guess for me, the intellectual breaking point sometime near the end of the decade of the 70s.
As Kenyon reevaluated his theory, new biochemical discoveries further weakened his conviction that amino acids could have organized themselves into proteins.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences.
Faced with mounting difficulties in his own theory and a growing body of scientific data about the importance of DNA, Kenyon was forced to confront the absolute necessity of genetic information.
The more I thought about the enormous problem that all of us who worked on this field had neglected to address, the problem of the origin of genetic information itself, then I really had to reassess my whole position regarding origins.
For Dean Kenyon, a new question became the focus of his search for life's origin. What was the source of the biological information in DNA?
If one could get at the origin of the messages, the encoded messages within the living machinery, then you would really be on to something far more intellectually satisfying than this chemical evolution theory.
Yet Kenyon faced a narrowing set of options, for he realized that as with pure chance, natural selection was also limited in its creative power. By definition, natural selection could not have functioned before the existence of the first living cell, for it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
Natural selection, chance, and his own theory of self-organization had all failed to explain the origin of genetic information. Now Kenyon saw only one alternative.
We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life was immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries of molecular biology.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
Amazing. Hopefully you all see how amazing our bodies are, and that all happens within the cell, okay? So he was talking about DNA or genetics, okay? So part of every cell that contains a genetic code, there are millions of component parts called nucleides, okay? And they have to arrange in a very specific sequence like what we saw in the video. And similar to like what we talked about just a few minutes ago. This is computer software, and it makes the computer operate and function computer software does. Well, the cell is the same thing. This is like the software of the cell. That's kind of like what we're talking about here. The DNA functions and orchestrates in a way that all of the cell and all of its growth can replicate with astounding speed. What we just saw happen, how that thing unwound and replicated and then went to another process, that happens so fast it's unbelievable. Unbelievable. Happens trillions of times, and that's within each cell in our body. And it can't have a bad day at all. It can't mess up. When a baby is conceived, it has to be error-free. A Christian named Francis Collins, he's in charge of the human genome concept, and this is where the order of genes and the mapping of them, and what happens if it's just a little bit off, okay? Which part is the DNA, is the gene in the DNA picture? What part is that? What is the gene in that picture, the double helix? What is the gene? Which part? Anybody know? I mean, it looks like a ladder, so what part of the ladder is the gene? It's the cross-section, or the rung, okay? So the rung of the ladder is called the gene. And if two pairs are off, out of billions of them, just two pairs, it can cause cystic fibrosis. So it can't mess up. It can't just randomly happen and these things get scrambled. Because you'll have, like we talked about mutations take away from, it will mess up. It won't be positive, it'll be negative. Just two pairs off can cause cystic fibrosis. Let's look at this from a little different perspective, because the DNA is also about language, okay? If you saw a message written in the sky that said, "Hello there," would you assume somebody wrote that? Well, you know somebody wrote that. You wouldn't think that it just popped up there on its own. Wow, check that out. Hello there. That just happened to form by clouds on its own. No, we wouldn't think that at all. You'd know it. Or like alphabets. If you eat alphabets and beside your cereal bowl in the morning it said, "Good morning," would you think, all right, Mom and Dad's playing a trick on me or something? Those things didn't just line up by themselves to say good morning all on their own. You know somebody did that. Well, the genetic code in a microscopic single-cell organism, if it were spelled out in English, it would equal the whole volume of Encyclopedia Britannica. That's a lot of words, a whole lot. And that's how much genetic code there is, similar to software code. Why would we think this could happen randomly? That's kind of crazy thinking, to even think that. So when you were in your mother's womb, the size of a dot, we could go in and retrieve the DNA if we could. At that point, anybody have any idea how long the DNA would be? Just when you were a dot, how long would that be? Have an idea? It would be 6 feet tall if it were to be stretched out. If that were to be stretched out when you were just the size of a dot, it would be 6 feet tall. And it would have on it encoded precise instructions for piecing together every part of your body. 600 muscles to 2 million optic nerves to 100 billion nerve cells. Every single cell in your body contains the entire blueprint for you. It's pretty amazing. And that really is an understatement to describe this. A newly conceived child is not a blob of protoplasm, as so many people still think, used to think and still think. Okay? It is not, so keep that in mind. And the two videos we just saw clearly show that it is not. Pretty amazing, God's design. A teaspoon of DNA, hard drives today. Can hard drives hold a lot of information like what's in this laptop or others? A whole lot of information, okay? They can store a lot, but nothing like a teaspoon, just a teaspoon of DNA. It will contain all of the information of every species on the planet, from 600 to 1,000 million species. A teaspoon of DNA, that's a lot of information. Also, that's not all, also every word from every book ever written. That's in 1 teaspoon of DNA, okay? In your body, it's been calculated that there's enough DNA that it would be more information than the entire internet, just in your body alone. An incredible amount of information, okay? A divine computer program requires a programmer, all right? That's why I gave this statistic, this one right here, cell by random chance. Sir Fred Hoyle, week 1 we gave this one. Sir Fred Hoyle, scientist, professor at Cambridge, and world-famous astrophysicist Chandra Wickremesinghe calculated for the probabilities of a single cell, this one, coming to existence as 1 in 10 to the 40,000th power in years. That's a whole lot more than billions of years. So in other words, what we're saying is it's literally impossible to do. Okay? It requires a creator. As I mentioned at the start of this class, how Christianity is challenged, and we do have answers in the defense of our faith, but realize our faith is the only one that can give answers like this, and adequate answers on top of that. And that's what we've been studying for the last 4 weeks. Actually, the last 9 weeks if we go back to the fall time in the first section, okay? Just realize this is more evidence for the Bible and its reliability. More evidence, okay? A lot of times we like to look to archaeology and prophecies and things like that for evidence for the Bible, and those are great and right. Science itself is more evidence for the Bible and its reliability.